Hello again, everyone, and welcome. This presentation marks the fifth activity of the fourth workshop within the MOOC program, and I'm pleased to introduce this educational pedagogical video on the scientific methodology and techniques for studying living beings, SMT-SLB, course. This video is an integral part of a training program specifically designed for newly recruited assistant professors, Class B, and it is supervised by the University of Mentouri Brothers, Constantine. To continue the introduction, my name is Dr SLAMA Kheira, representing Dilali Bunama University of Chemis Miliana. I am part of the Faculty of Natural and Life Sciences, specifically within the Department of Biology. This pedagogical video is relevant for the upcoming 2024 to 2025 academic year. Educational video content. This educational video, specifically tailored for second-year biological science students, focusing on scientific methodology and techniques for studying living beings, or SMT-SLB. In this lesson, we will explore the essential methodologies and advanced techniques crucial for investigating living organisms at the cellular, molecular, and genetic levels. Our video content will guide you through several key areas. First, we'll explore cell morphology studies, where you'll discover techniques like photonic, light, microscopy, and electron microscopy to observe cellular structures in detail. Next, we'll delve into biochemical composition analysis, learning methods such as chromatography, electrophoresis, and immunocytology to analyze various cellular components. Then, we will dive deep into genetic engineering techniques. This includes exploring DNA extraction, PCR, RT-PCR, and DNA sequencing, including methods like the Sanger method, to manipulate and study genetic material. Finally, these powerful techniques are extensively applied in modern biological research, medical diagnostics, and biotechnology. Course summary. This course is fundamentally about applying scientific methodologies and advanced technologies to study living organisms at cellular, molecular, and genetic levels. We will cover essential techniques that will significantly enhance your research capabilities and decision-making in biological sciences. These include vital tools such as microscopy, chromatography, electrophoresis, and comprehensive DNA analysis. Through modern teaching approaches, students will possess comprehensive knowledge to effectively understand, develop, and apply these methods. The curriculum is thoughtfully designed to integrate theoretical foundations with hands-on practical applications. This approach aims to thoroughly prepare students to contribute meaningfully to breakthroughs in life sciences, medicine, and biotechnology. By the end of this course, learners will acquire the skills to conduct rigorous scientific investigations and drive innovation in biological research. Concept map of SMT-SLB course. On this slide, we have a comprehensive concept map that visually outlines the intricate structure and rich content of our scientific methodology and techniques for studying living beings, SMT-SLB, course. This map is meticulously designed to give you a clear, interconnected overview of how the different methodologies and techniques are organized and will be explored throughout the program. Course content. The course content is thoughtfully structured and divided into several distinct learning units, each meticulously designed to build upon the last, ensuring a thorough and progressive understanding of the subject matter. Unit 1. Introduction and Course Overview. We will begin with a foundational introduction to scientific methodology for studying living organisms. This unit will clearly outline the course objectives, its overall structure, and the key concepts we'll explore throughout the program. You'll gain a clear understanding of how we'll approach biological research techniques. Unit 2. Methods for studying cell morphology. Here, we will dive into various microscopic techniques. This includes starting with photonic microscopy methods like bright field, phase contrast, and fluorescence microscopy, and then progressing to advanced electron microscopy, covering both transmission electron microscopy, TEM, and scanning electron microscopy, SEM. Practical exercises will help you determine the best microscopic approach for different research needs. Unit 3. Biochemical composition analysis. In this unit, our focus shifts to techniques used to study the intricate components of cells. We'll cover chromatography for separation, electrophoresis for analysis, and spectrophotometry for precise measurement. You'll also learn immunological methods for antigen localization, with exercises designed to test your understanding of these biochemical techniques. Unit 4, Genetic Engineering Techniques. We will then progress to powerful molecular methods. This begins with DNA extraction and purification, followed by an exploration of PCR amplification and both manual and automated DNA sequencing. We will also engage in interactive exercises, such as matching techniques to their specific applications and explaining key principles like phase contrast microscopy. Implementation process. The implementation process is essentially about how we'll actively engage with and apply the knowledge gained in this course. In this phase, students will apply the knowledge gained from lectures through guided practical sessions. Working in pairs, each team will conduct workshops, research presentations, and lab sessions, covering different course topics. We have structured the implementation around 4 key areas. 1. Interactive workshops. These are guided practical sessions where you'll get hands-on training with essential equipment like microscopes, electrophoresis apparatus, and PCR machines. We'll provide guided protocol execution with continuous instructor feedback, and you'll learn to troubleshoot common experimental challenges that arise in the lab. 2. Research presentations. In these sessions, working in pairs, each team will present applied research projects, for example, comparing different microscopy techniques. We will facilitate structured peer discussions, guided by clear evaluation rubrics. They'll also participate in a journal club format for analyzing published research papers. 3. Lab practicums. Our lab practicums are dynamic, involving rotating through stations for core techniques such as cell imaging, comparing phase contrast versus fluorescence, DNA extraction quantification, and protein separation analysis. Crucially, your lab notebooks will be graded for scientific rigor. 4. Assessment includes, beyond the specific activities, our assessment within this process includes technical competency evaluations, research poster presentations, and lab report peer review sessions. Teaching and learning activities. Our teaching and learning framework is specifically designed to ensure active, documented, and applied learning throughout the entire scientific methodology course. This approach is built on 4 key pillars to maximize students' engagement and skill development. 1. Active participation requirement. The mandatory attendance for all lectures, workshops, and lab sessions is required. This ensures regular engagement in both the theoretical and practical components of the course. 2. Comprehensive note-taking. To reinforce learning, students are required to systematically document 3 key areas: all experimental protocols, the various research methodologies we cover, and the key scientific concepts discussed in class. 3. Interactive learning approach. We will foster an interactive environment through structured Q&A sessions after each technique demonstration. We'll also utilize peer discussion forums for collaborative problem solving and engage in case study analyses of published research to connect theory to real-world applications. 4. Applied knowledge integration. This final pillar focuses on putting students' knowledge into practice. They will be implementing the techniques they learn through hands-on laboratory experiments, developing and presenting their own research, and engaging in exercises focused on data interpretation. Assessment system. The assessment system is designed to be comprehensive, ensuring that students' final grade is a fair reflection of their continuous progress and overall mastery of the course material. It is composed of 2 main components, both of which are equally weighted at 20 points. First, we have continuous assessment. This ongoing evaluation tracks your learning throughout the semester and is divided into 2 parts. Knowledge checks, worth 5 points. This includes short quizzes and reflection journals, which will often contain synthesis questions to test your deeper understanding. Projects and practical evaluations, worth 15 points. This section encompasses a variety of practical work, including group research project, 4 points, an individual case study, 3 points, lab reports, 4 points, and a final practical exam, 4 points. The second component is the final examination. This is also divided into 2 parts to evaluate both your theoretical and practical knowledge. Written component, worth 15 points. This part includes problem-solving questions, 8 points, synthesis questions, 5 points, and multiple choice questions, 2 points. Applied component, 5 points. This section focuses on students' abilities to apply their knowledge to real-world scenarios, including an experimental design and a data interpretation exercise.